Greetings to you all. This is TR.Dr. MUGOMBA Ronald. I'm back again, and we are going to go ahead and look at the process of DNA replication. That is what we are going to go ahead and look at, DNA replication. Consider if this is your first time to this YouTube channel, consider to like, consider to share, consider to subscribe, because this is the only way you can support our YouTube channel, so that you can encourage us the more to continue making for you better and good videos that will enable you to successfully study biology with ease. So we are going to go ahead and look at the DNA replication today, and this is teacher Dr. MUGOMBA Ronald, MBCHB, BSE. Good. So let's get started today. When you look at DNA replication, we usually see DNA. The purpose is that the purpose of this process known as DNA replication is we usually want to make copies of the DNA, and the reason as to why we need to make copies of DNA is simply because cells usually undergo a process known as cell division. So since cells usually undergo the process of cell division, we ought to go ahead and make sure that we make copies of the DNA before replication is done, before cell division is done. Otherwise, if we are to divide the cell continuously without replicating genetic material, we would have each cell having less by half the amount of genetic material, less than half, or half every generation, half that of the parent. So to avoid that disruption on the amount or the content of genetic material in each of the cells, usually we have to undergo DNA replication so that we can ensure successful cell division to take place. Now, there are a number of mechanisms or theories that have been put forward to try to explain how DNA replicates. So we need to go ahead and look at each of them. So a number of theories. The first theory that was put forward is what they usually call the we have what we usually call the semi-conservative model of DNA replication, where we see that the parent DNA is usually used. It is unwound, and each of the parent strands is used as a template to form new DNA or to form daughter DNA. And at every point in time, you realize that, as you can see clearly here, that this was the parent DNA which divided, so it unwound, and then we formed daughter DNA, daughter strands. So in the second generation, you can all attest with me that you can see that in the second generation, the second generation of the DNA is made up of both the parent strand and the daughter strand, and so on and so forth. So at every replication, we usually see one strand of the parent being retained in each of the DNA, plus a new strand of DNA in the daughter genetic material. We have a parent strand being maintained along with a new strand. That is semi-conservative model. In conservative model, we usually see that we only form all the genetic material of the parent is maintained in one copy, and all new DNA is formed in another copy. So in conservative, the whole DNA of the parent is conserved in the daughter cells, and so one cell ends up having the daughter cell has completely new one of the daughter cell has new genetic material, then the other one has the parental genetic material. That is conservative. Then we also have what we usually call dispersive model, So we shall not put so much attention to this passive model. There are a number of experiments that were conducted to go ahead and confirm whether DNA really replicates through conservative or semi-conservative model. Now, these experiments were suggested by, they were conducted by a man known as Michelson and Starch experiments. So they conducted experiments to confirm whether DNA replication takes place through conservative model or non-conservative or semi-conservative model. So what they did is they cultured bacteria in a medium that contains heavy nitrogen, as you can see from here. So they cultured the bacteria in a medium containing heavy nitrogen for some time. After some time, they transfer this bacteria in a medium that contains light nitrogen. And after some time, they went ahead, after the transfer, after 20 minutes, they went ahead and centrifuged this bacteria, or the content of the bacteria, and they observed. So after 20 minutes, they went ahead and centrifuged the contents so that they can ascertain the composition of the DNA in the bacteria that is present. And then after 40 minutes, they once again centrifuged. This confirmed the semi-conservative model. In the first 20 minutes, they found out that it is made up of both heavy bacteria and light bacteria. And in contrast, so when they found out in 20 minutes, centrifuge experiments showed that it was a 50 by 50 of both light nitrogen and heavy nitrogen. It was not as expected that in the conservative model they expected to have light nitrogen having a certain being centrifuged at a different area, and then heavy nitrogen being centrifuged at the bottom. That didn't happen. They had combination, they had a mixture of the two being centrifuged at the same level in the first 20 minutes. That is to say, both light and heavy at the same level. It's a mixture. It is an intermediate. That's what they observed, as you can see from here. Then after 40 minutes, they observed that they had both the light nitrogen DNA at a higher level plus a mixture. So you can see, instead of having clearly light the way we are seeing it here and then heavy distinctly separated, in the second generation, I would say they had both light at the top, and then they had the one that is a mixture. Instead of having light and heavy, they had the one which was a mixture of light and heavy, centrifuging at another different level. So that confirmed that when you look at DNA replication is usually taking place through a semi-conservative mode. So that confirmed. So DNA replication never takes place through non-conservative or through conservative model, or it only takes place through the semi-conservative model, whereby in each of the cells formed, the daughter cells, the DNA is made up of both the parent and it is made up of both the parent strand and the daughter strand. So it is a combination of two strands. That is the semi-conservative model of DNA replication, as you can see it from this illustration that we are having here. In semi-conservative model of replication, we are seeing that the parent strand is maintained. Along with the parent strand, we synthesize a daughter or a new strand. So the daughter cells will have 50% of the parent DNA and 50% of new DNA. So that is what I can say. And as generations go by, we shall see it goes like that, it goes like that. That is why in the first generation they had a mixture of 50% parent and 50% heavy and 50% light. And as time went on, light started dominating. 75% light, 25 heavy, like that. Instead of completely disappearing, the heavy one completely disappearing at once. Now, let us go into the details of the whole process. So base pairing allows each of the strands to serve as a template for the new DNA. Each of the strands in the parent DNA will act as a template, and we shall synthesize new DNA using each of them, as you can see from there. Now, new strand is usually half the parent and half the new DNA, and we have seen that already. So now we go ahead and look at the DNA material, or when you look at the DNA structure, you'll all appreciate with me that DNA structure is always considered to be what we usually call it is bonded from phosphates to sugar between a 3' end and a 5' carbon. And you realize that this DNA molecule has direction. That is to say, it moves, you can, as you read, for you as a person, you might read it from the 5' to 3' direction. But you can see clearly that it has direction. The 5' being the side which has the phosphate group, and the 3' which is having the hydroxyl group projecting outwards. So you remember always that the 5' end is the one that is having the phosphate group projecting outwards. Then the 3' end is the one that has the hydroxyl group pointing outwards. Then, so they're usually complementary. Whenever we are making pairs, whenever we are pairing to form a double strand, they're usually complementary. We usually follow the base pairing rule, whereby we usually pair the purines with the pyrimidines using a complementary base pairing rule, which usually does not allow us to bind adenine. So we usually bind adenine with cytosine. Sorry, adenine with thiamine. Then we always we usually combine cytosine with guanine. So that is the base pairing rule, and you will all appreciate with me that they're usually anti-parallel to each other. What do we mean when we say anti-parallel to each other? We mean if one strand is moving in the 5' to 3' direction, as you can see here, the synthesized strand that is being synthesized, another strand will be moving from 3' to 5' direction. That is the meaning of what we call the anti-parallel nature of DNA. It is anti-parallel. That is to say, these strands move in opposite direction to each other. If one is moving 3' to 5', the other one is moving 5' to 3'. You can all see that. So we have 5' to 3', then we have 3' to 5'. So that is the basic structure. So we can go ahead and look at the different types of bonds that are forming the DNA. So DNA has both strong and weak bonds holding it. So we can go. We shall see how each of these bonds fit in when it comes to bonding of the DNA. So we have the first bond that is known as the hydrogen bonds, which are usually present between the nitrogen bases. But you can all notice with me that we have between thiamine and adenine, we have only 2 hydrogen bonds, as you can see from here. Then we have between guanine and cytosine, we have 3 hydrogen bonds. That makes gyase bonds stronger than the TA bonds. Always note that, very important. So even when we are looking out for sites where we can easily open up the DNA, the easiest site to open up the DNA would be the TA site as compared to CG site, because CG site will be stronger and more resistant to being opened up. Now, having looked at that, that takes us to our next type of bond. We have covalent bonds. So you can see the phosphodiester bonds. The phosphodiester bonds being formed between the 5' and the 3' carbon, and they are held in place by the phosphate making a bond with the hydroxyl group. So that is the phosphodiester bonds. They are covalent and they are stronger than the hydrogen bonds. In fact, when we are looking at DNA replication, when we are breaking the bonds that we usually break to allow DNA replication to take place are usually hydrogen bonds, as in contrast to covalent bonds. So when you look at DNA replication involves a very big team of enzymes which all coordinate. There are about 12 different types of enzymes which all coordinate together to bring about the process of replication. So on the chromosome, we usually have what we call the origin of replication. So usually we form an origin of replication, and in eukaryotic cells we have more than one origin of replication. So DNA replication can start from different origins, different areas within the so we can have very many different origins of replication. That is why sometimes eukaryotic DNA is usually considered to be polycistronic, because one DNA can be used to synthesize very many different other DNA at the same time. So we can have replication taking place at different points. We can start from different points on the DNA, unlike the bacteria where they only have one origin of replication. So we have many origins of replication when it comes to the eukaryotic DNA. So you can see clearly there is an illustration here that is showing you how the replication forks look like, and how the replication bubbles or the origin of replication looks like under the light microscope. Okay, now let us go ahead and look at the whole process of replication and how it takes place. So the first step of replication all begins by the action of unwinding the DNA. How do we unwind the DNA? There is an enzyme that we are calling what they call helicase enzyme. So helicase enzyme here is the enzyme that plays a very important role when it comes to breaking down the hydrogen bonds that exist between the DNA. So the helicase enzyme will come and then unwind the DNA. So after unwinding the DNA, these nucleotides have a tendency, they are sticky, so they have a tendency to rewind with each other. So to prevent rewinding with each other, we usually have what we usually call, so we usually have so the helicase enzyme is very important when it comes to unwinding the DNA helix. It unwinds in both directions, and it creates what we call the replication fork. Now, the unwound or these single strands of DNA now are stabilized by what we usually call single-stranded binding proteins. These single-stranded binding proteins are very important because, one, they prevent this DNA from rewinding, because they're usually sticky, so they have a tendency to rewind again. And two, it also protects the DNA from being worked on by endonucleases. Otherwise, if you leave it open like that without those single-stranded DNA, one, it is most likely to reform again, so the molecule will close. Then two, it might be worked on by endonucleases, which might cause mutations, so those enzymes might start cutting it down before we replicate. So then we have another enzyme known as DNA ligase. So gyase, DNA gyase, sometimes this DNA gyase is sometimes known as topoisomerase. Topoisomerase enzyme is very important in preventing supercoiling upstream. You know, as some of you who have opened up a rope, if you try to open up the rope, you realize that you can create a tension or a torsion up the stream. So as you unwind, there is a tendency that up the stream, like for instance this side, as you unwind this region here, this side might become very tense and it will resist unwinding, and that torsion there will prevent the process of DNA replication from taking place. So it will harden the work of helicase. So what topoisomerases do, or what they are calling DNA gyase enzymes, what they usually do is they cut certain areas. They have two components. They have hydrolase component or they have what they call ligase component, which comes and cuts this side. It cuts. After cutting, then it joins again. It cuts and then joins, making it very easy for the helicase to work. So it prevents supercoils. It goes ahead and prevents supercoils upstream, or prevent tangling of the DNA upstream. As the helicase is moving, it will find it very easy to open it up. So it is sometimes known as topoisomerase. It has two components. It has the ligase component, which cuts the DNA upstream, and then it has the ligase component that goes ahead and joins again. So it prevents torsion upstream, or it prevents supercoils, or what they call the tangling up the stream. That is known as DNA topoisomerase, or sometimes known as gyase, DNA gyase. Good. So we have seen the single-stranded DNA. We have seen the replication fork and how it is formed by helicase enzyme, because we have said helicase enzyme is very important when it comes to unwinding the DNA. Then the single-stranded DNA is very important. The single-stranded protein is very important when it comes to it prevents the closing of the DNA strand, and also protects this DNA from being digested by the endonucleases. Now, we have also looked at the topoisomerase or the gyase enzymes, which are very important in preventing supercoils upstream, or in preventing tangling of the DNA upstream. So our second step is going to be we have to prime. As before we can form the new DNA strand on the parent strand, we need to start by priming the DNA. So we have to prime it. We usually do priming of this DNA, priming of the strand, because this enzyme known as DNA polymerase 3, we will realize that we have a number of different examples of polymerases. The first enzyme that we usually use when it comes to adding nucleotides, new nucleotides, DNA nucleotides, to form the new strand of DNA. As we read this strand from the 3' to 5' direction, we are going to synthesize the new strand in the 5' to 3' direction. That is how the DNA polymerase 3 synthesizes. But for it cannot synthesize, it cannot add nucleotides. It cannot add nucleotides when they don't have a primer. So DNA polymerase 3 has a tendency that it has to combine nucleotides to a 3' end of the nucleotide. That means that it has to add to some chain that is already growing. So what usually happens is RNA primase comes and adds its own. It first forms RNA primers. RNA primers. They are simply nucleotides of RNA, so that it can form a beginning point for DNA polymerase to start working. So now, after the primers have been added by RNA polymerase, then it creates the 3' end onto which this DNA polymerase 3 is going to add. Because it has a tendency, DNA polymerase can only build new DNA only to nucleotides which are existing on the DNA strand, and specifically to the 3' end of the nucleotide. So now we start. So after priming, now the next step is going to be DNA replication. Now in DNA replication, DNA polymerase is going to come in. We are building daughter DNA strands, and we are doing it so we add new complementary bases. We get them from the cytoplasm, and they are imported into the nucleus. Then we use the base pairing rule to add the different nucleotides, and this is done by DNA polymerase 3. DNA polymerase 3 is the one that does that job of adding nucleotides, new complementary bases onto the 3' end of the nucleotides. So it is building. So that is DNA polymerase. It's very important. It is building the new strand from the 5' end to the 3' end direction. 5' to 3' direction. It reads the old strand in 3' to 5' direction. After reading it, it synthesizes new strand from 5' to 3' direction. So now you can see how it is being constructed in an antiparallel nature. And you can see we are synthesizing new DNA. So that is how we are able to synthesize. So after that is done, after that is done, then the next step is we are going to go ahead and replace these primers, the RNA primers, by DNA. Remember, the RNA primers were put simply because so that we can have a starting point upon which DNA polymerase 3 can start working. So after we have synthesized our new DNA strand, we have to go ahead to replace those RNA nucleotides with DNA nucleotides. How is that done? It is done by the use of an enzyme known as DNA polymerase 1. So DNA polymerase 1 now comes in to facilitate the removal of the primers. The binding of the primers or the fixing of the primers was done by RNA primase, so that it can create a starting point for DNA polymerase 3. So DNA polymerase 1, you can clearly see that it is done that is going to facilitate removal of the primers. So now we need to understand some concept here. When you look at DNA replication, you realize that we are using both strands of DNA. That is to say, we are using the DNA that is being read from the 3' end to the 5' end direction, and then we are also using the strand that is being read from the 5' end to 3' end, all of them as templates. So we are using them, all of them, as templates to form DNA. So as Helicase comes and opens them up, what we usually do is start opening. So we start seeing the growing helication fork. The growing replication fork is moving in that direction. So we have the 3', we have the DNA strand that is starting from the 3' end, you can see it here, to the 5' end. Then we have the one which is starting from the 5' end to the 3' end. But we should remember very well that our DNA polymerase 3 reads 3' to 5' and synthesizes 5' to 3'. Okay, so there we are. So when you look at DNA polymerase 3, for it, when it is reading, it reads 3' to 5', and then it starts synthesizing DNA in 5' direction to 3' direction. Okay, so this DNA polymerase, so we shall have two types of strands which are being formed. New DNA strands will be in two categories. We shall have the leading strand and the lagging strand. How do we distinguish between the leading strand and the lagging strand? The leading strand is usually complementary to the DNA strand that is 5' to 3' direction. That is usually the one they call the leading strand, and it is complementary to the 5' to 3' direction. And another way you can remember it is the fact that for it is made continuously. In its 5' to 3' direction, it is continuous and it does not have Okazaki fragments. And then the first one I told you, you can identify it by looking at its what it is complementary to. It is complementary to the DNA which is 5' to 3'. Sorry, it is complementary to DNA that is 5' to 3'. Okay. Or what we can say, it is complementary to the template strand which is read in the 3' to 5' direction. Anyway, the reading is usually 3' to 5' direction, but according to what we are having here, for it is going to be read in the 5'. It is complementary to 5' to 3'. Okay. So that is what they call the leading strand. It is read. It is constructed continuously. That is how it is constructed. Constructed continuously, and it is complementary to them 5', 3'. strand. Now we have another known as the lagging strand. This lagging strand, yes, it is also synthesized in a 5' to 3' direction, but you realize that for it, so someone might think that we have some kind of synthesis of 3' to 5' direction. That never happens. DNA polymerase 3 cannot synthesize nucleotides, cannot synthesize new DNA using 3' to 5' direction, but it can synthesize DNA only in the 5' to 3' direction. That is how it is able to synthesize. So how does the lagging strand work? So for it, it is going to be complementary to the parent DNA, which is 3' to 5'. So what happens is it synthesizes DNA. It is synthesized in small sections. So you will realize that for it, when you observe it, it will have very many RNA primers. You can see. For it, it is synthesized in a discontinuous manner. It is synthesized in a discontinuous manner, and it is complemented to the 3', 5' direction, 5' DNA, and then 3, it has very many primers, RNA primers. So it limits DNA polymerase. When you look at limits of the DNA polymerase 3, it can only build onto a 3' end of the existing DNA. We saw that already. So we end up forming what we call the Okazaki fragments in the lagging strand. The leading strand does not have Okazaki fragments. The leading strand is formed continuously. Lagging strand is formed discontinuously. Leading strand does not have, it has few primers. Lagging strand has very many primers. Leading strand is complemented to the DNA strand known as 3', 5', 3'. Or if we are to read it from this side, the leading strand is complemented to the 3', 5', the 5' parent strand or parent strand or template strand. Then the lagging strand is complemented to the 5' to 3'. But the reading of DNA by DNA polymerase, it reads it from the 3' end to 5' end, and it synthesizes in the 5' to 3' direction. These fragments were first observed by a man known as Okazaki, a Japanese, who first observed them, and that is why they were named after him. So what usually happens, so when you look at Okazaki fragments, they are rejoined again. We first use enzymes which are going to go ahead and slice out the primers, the RNA primers. After they have sliced them out, then ligase comes in to join those Okazaki fragments to form a continuous DNA strand. Okay, so we have seen that leading strand is synthesized continuously. Lagging strand has Okazaki fragments and joined by a gyase spot welder enzyme. So it is a spot welder enzyme. Slices out the RNA primers and then joins the DNA strands. So DNA replication on the lagging strand, when you look at it, you can see a primase. We saw that after the primase has put RNA primers, then DNA polymerase starts synthesizing very well the new DNA strand, but by adding at the 3' end of the RNA nucleotides, the hydroxyl group. Then when you look at the lagging strand, how it is formed discontinuously. You can see that the primer is added and then we form DNA. DNA nucleotides are also added in that way. So RNA primers are added by primase enzyme. We saw that already. However, short segments called Okazaki fragments are usually made because it can only go in the 5' to 3' direction. Otherwise, if it could form DNA strand in the a 3' to 5' direction, we wouldn't form Okazaki fragments. So because DNA polymerase can only synthesize 5' to 3' direction, that is why we end up with Okazaki fragments. So after all is done, ligase comes in and welds these Okazaki fragments together by getting rid of the numerase. You can see the numerase primers, RNA primers. So they are removed. They are removed by an enzyme known as DNA polymerase 1, and then ligase comes and welds the Okazaki fragments together. So polymerase 1 comes and then slices out the primers. Then the ligase comes and welds. You can see that very well. It is welding and removing. It's welding them very well. So the next DNA, so we have seen all that. So the strands are glued together by the DNA ligase. Okay, so we can now have a recap. As we conclude, we can have a recap of what we have been looking at. We started by the direction. The DNA polymerase reads in the 3' to 5' direction. After reading so, it synthesizes new DNA in the 5' to 3' direction. The helicase is done that opens up the DNA double helix. After that is done, the small nuclear proteins go ahead and bind. They bind on the single strands. Single-stranded DNA, single-stranded protein binds on the single strands and prevents them from sticking back or rewinding, and also prevents them from the activity of endonucleases. Single-stranded, and then we saw primase. Primase, very important when it comes to establishing a starting point for polymerase 3, creating the hydroxyl group, the hydroxyl end onto which polymerase 3 can add DNA nucleotides. DNA polymerase now starts constructing the new DNA strand. Then we see Okazaki fragments in the lagging strand, and then we see DNA ligase coming in to join those Okazaki fragments, but it does it along with the DNA polymerase, which removes or excises the primers. So the leading strand has few primers. I would say I wouldn't love to say it is one, because we have numerous lubrication bubbles. So that is how the whole process is known as DNA replication takes place. I remain teacher Dr. MUGOMBA Ronald. If you've enjoyed the video, please consider to subscribe, comment, share with your colleagues, because that is the only way you can go ahead and encourage us to continue making good videos to make biology simple for you. I remain teacher Dr. MUGOMBA Ronald. Have a nice day ahead of you. Till next time.